Good evening, everyone. My name is Sam Hoggett, and I am the Chancellor of the University of California, San Francisco. And it's my great pleasure to welcome you all here tonight for the first in a series of 3 weekly panel sessions to mark what has now been almost a year from when the World Health Organization declared a global pandemic due to the novel coronavirus, SARS-CoV-2, and the tremendous impact that the disease caused by this virus, COVID-19, has wreaked across the globe in the last year. Hundreds of millions of people have been afflicted by the disease, and more than 2.5 million have lost their lives. This is a horrendous tally in just a year wreaked by the pandemic. But tonight, we're going to focus on the science that will drive recovery from this disease. In just over a year, we have all gone from first learning about a novel virus emerging across the globe to a fundamental understanding of the biology of this coronavirus, understanding how to test for it, how to sequence the genome, how to follow its passage from population to population around the globe, how to manage patients who are afflicted with the disease COVID-19, how to conduct clinical trials during a pandemic, how to discover new therapies, and ultimately preventive vaccines for this devastating disease. The science throughout the last 12 months has been truly breathtaking. And it's my great pleasure tonight to introduce a panel of illustrious scientists from the Bay Area who will discuss these remarkable advances. Tonight, we have with us Jennifer Doudna, Professor of Biochemistry and Molecular Biology at UC Berkeley, and also with appointments at UCSF and at the Gladstone Institutes. Jennifer was the winner of the 2020 Nobel Prize in Chemistry. Joining her will be Joe DeRisi, Professor of Biochemistry and Biophysics at UCSF and co-president of the Chan Zuckerberg Biohub. Joel Ernst, Professor of Medicine and Chief of the Division of Experimental Medicine at UCSF, along with Annie Lukemeyer, Professor of Infectious Diseases at UCSF, and finally, Ashish Manglik, Assistant Professor of Pharmaceutical Chemistry at UCSF. Moderating the session tonight will be Dan Lowenstein, Executive Vice Chancellor and Provost at UCSF. And now I'll turn it over to Dan to moderate the session. Thank you for joining us.
Thank you, Chancellor Hoggett, and welcome everyone to this, this focus on how science has had such an impact on our lives. Just to reiterate what the Chancellor just reviewed, just stepping back for a moment and thinking about what an extraordinary, incredible year this has been. Amazing to think that 2 days from now will mark the anniversary of the WHO declaring a worldwide pandemic, the millions of lives lost, the incalculable economic fallout, and the way it's shattered virtually everyone's lives across the planet. But as has been the case in the past in history, despite these enormous hardships, we have so many reasons for hope and a better future now. And it is truly incredible to realize that we've gone from a lack of an understanding of what this virus could do to us at the onset to now having in hand 3 highly effective vaccines, which is just an extraordinary achievement. And as you're going to learn from the discussion from these eminent scientists that have been brought together for this panel, this didn't all just happen in 1 year. It's built upon a legacy of biomedical research and innovation and creativity that benefits all of us in society. So tonight we're going to have a conversation, and we're going to focus in on a couple of different areas within biomedical research. We'll talk about the technologies that have helped us identify the novel coronavirus. We're going to talk about how we have methodically tested potential treatments in the midst of a pandemic, what some of the therapies for infectious disease that may emerge in the future, and what tools we can use to help detect and respond to future dangerous outbreaks, because these outbreaks will continue to occur. I think we all agree that we're better prepared than ever to handle the next one that comes along. So with that, let's launch into our discussion. So I'm going to start, first of all, and because I know you all, I'm going to refer to you by first names, these doctors. So first of all, Jennifer and Joe, let me ask you the first question, and that is, starting with you, Jennifer, how did the technologies emerge over the past decade that have led to our ability to rapidly identify SARS-CoV-2 and to come up with this effective response? Would this have been possible a decade ago?
I think it would not have been possible a decade ago. And it's truly extraordinary to appreciate the advance that we have seen in real time over the last year that, as you said, as Chancellor Hoggett mentioned, pulls together all the work that's been done in the previous decades to understand virology, understand how to sequence rapidly, nucleic acids, and understand what they encode, solve structures of viruses. That was done very quickly for this coronavirus as well. And to then start to understand how viruses interact with their hosts, because that's, again, been something that has been incredibly enabling. And a lot of that work has been done at UCSF, as you know. So I think this is really a moment. It's an extraordinary moment. On the one hand, terrible tragedy. On the other hand, incredible triumph of science that we now have 3 wonderful vaccines and more on the horizon that are going to lead us out of the pandemic and hopefully prepare us better for future pandemics.
Mm-hmm. And Joe, I mean, you are known as a virus hunter, so you've been at this for quite a while. How about you? What do you see in terms of the technologies and the pace of development of these tools?
Yeah, I'll just build on what Jennifer just said. We have 3 tremendous vaccines in hand, but that all starts with knowing what the virus is and knowing what it's made of, what its genetic code tells us. And to do that, we need to read the genetic sequence of the virus. And if we look back, go back some number of decades to the 80s, it took years to figure out that HIV was the cause of AIDS and to read its genetic code. This could be done in a matter of days now. And that power, which has come along so far in the last decade, has really catapulted efforts at both therapeutics, vaccines, the structural biology, understanding the shape of the proteins, as Jennifer alluded to. All that stems from knowing the sequence first. We have to be able to read the book, the genetic code. And that also has enabled us to track the virus, where it goes, what it's turning into, because it doesn't stay still. It's constantly evolving, mutating, as all viruses do. And that's really going to let us keep our finger on the pulse of the virus and the pandemic the whole time. One can argue we need to do a better job at that, and we could talk more about that later. But this technology has made possible what was unthinkable 15, 20 years ago.
So back to you, Jennifer. You're also a virus hunter of sorts, but you initially focused on the way viruses attack bacteria, and of course that was the basis of your remarkable discovery of CRISPR. And so you've been thinking a lot about how the structure of the molecules that Joe was just talking about, DNA, RNA, protein, how they recognize one another, how organisms recognize these molecules. So can you tell us a little bit more about how understanding the 3-dimensional structures of these things helps in understanding SARS-CoV-2 and what's on the horizon in terms of understanding that structure and coming up with therapies?
Right. Well, as Joe just said, the start of the whole effort is certainly to identify what this virus was, first of all, and secondly, to start thinking about how we would protect ourselves from it, started with getting the sequence of the genetic material of the virus. And fortunately, that sequence was released early by China back in January of last year. And immediately, almost immediately, companies as well as academics began combing over that sequence and trying to figure out, what it could tell us about how the virus worked. Quickly, structural biologists were able to determine structures of proteins that make up this virus, as well as ultimately the entire virus, and see what it looked like. And that told us that a protein known as the spike is interacting with a receptor on human cells. And that's really the lock and key mechanism that allows infection to occur. And so for folks that were developing vaccines, that immediately gave them a strategy to use for vaccination. The idea was, let's educate human cells, human bodies about this spike protein so they could harness and have an immune response that would then be present should the real virus show up. And that's effectively how the vaccines are all working. And that certainly would not have been possible without that combination of genetic sequencing and our knowledge of the structures and functions of the molecules that are involved in infection.
Mm-hmm. Mm-hmm. Well, let's shift to the way that the identification of the properties of the virus then led to the ability to think about treatments. In the acute phase of the pandemic, with people, dying so acutely from this powerful new form of a virus, we were thinking about how to save people in the ICU and what therapies we could initiate instantly, knowing that longer term vaccines were going to have to come along. So Annie, let me turn to you about the role of clinical research, clinical trials, in order to identify what drugs might work at the outset and what we've done since then. So to the outside world, I think to the public, the trials during the pandemic may have seemed a bit rushed. You're a physician scientist, you're a clinical trialist. Did they feel rushed? And what have we learned during the pandemic about conducting scientifically sound clinical trials during such an acute public health crisis?
Yeah, so thanks, Dan. And I can understand why that may be the perception. But here's what I'll say to that is that we needed to go quickly. And it's appropriate to go quickly. And there's a difference from being rushed and disorganized with moving quickly because we must. And anytime that I start to feel like this is too hard and it was overwhelming, I think about Ebola, and which is a very scary disease as well. And the remarkable work that they've been able to do in Ebola, including very rigorous clinical trials that have shown us that there's a vaccine and what the role of monoclonal antibodies are. So we can do this work in the setting of pandemics. It's hard. I will fully acknowledge that. And many of us have been doing that work and it's been challenging. But just because we had to move quickly, in the beginning, I think one of the biggest challenges was lack of coordination and a lot of small studies doing the same thing all over the place. But I think that really has come together. And unfortunately, in the very beginning, we were operating without any clinical trial data. So we needed to go quickly so that we could figure out what the best things to do and what we should stop doing. And the other thing just to keep in mind in the clinical trial world is that we expect there to be what we call negative studies. Not everything we study works. We always want everything to work, but it's very usual for us to study a lot of different agents. And when we find one that doesn't work, that's great news. That means that we can move on, we've learned something about this disease and how it works. And we often will do more than one study of the same drug or therapeutic to figure out, hey, are we really seeing what we think we're seeing? So we rarely just do one blockbuster study, say, great, this works, and then move on. So to the outside world, it may seem, wow, what are they doing? Because there's been such scrutiny of clinical trials. But I think we're just seeing a very compressed timeline of what really is the natural history of clinical trials, which is that we need to test a whole lot of things, which is really important, do it quickly, and many of our interventions aren't going to work, and that's really helpful to know.
Mm-hmm. Mm-hmm. And Annie, what about just the plain challenge of coming up with an effective therapeutic for this virus? I mean, we struggle to have completely effective and safe therapies for any viruses, but this one has also proved to be fairly intractable. What is it about these diseases that are so therapeutically challenging? And have we learned anything from the COVID experience thus far?
Yeah. So I would say that one of the challenges has been that how we approach this disease is different where you are in the disease stage. And that's really clear. So that people early in the disease, that some interventions work much better when people aren't critically ill. So probably the antivirals, we want to get those to people as soon as possible, because we know by day 10, we have trouble isolating culturable virus. So if you want an antiviral to work, we got to get to people quickly. That means we have to diagnose them and then link them to care and provide them with an antiviral that they can access. Other medications like the monoclonal antibodies are increasingly looking like, because they are antivirals, that they need to be given really quickly to make a difference. And when we're giving them later in disease, they haven't been so helpful. On the flip side, we know that when people become critically ill from COVID, that it can become quite an inflammatory state. And that while you may want to treat the virus that's there, you also may really benefit from giving anti-inflammatories like dexamethasone or baricitinib or some of the many other immune modulating strategies that we have. But if you give those too early, you actually may be causing harm. So I think we've really had to recognize that this isn't just a one size fits all, and that recognizing where people are in the stage and what the biology is informs how we treat people. The last thing I would say there too, Dan, is that the natural history of COVID is that most people get better, which is great news. But that's made it really confusing, because I could give someone a medicine that does absolutely nothing and most people are going to get better. And that's led to a whole lot of confusion when people say, but wait, look, we gave them this and 80% of people got better. It makes it all the more important that we do really rigorous studies. So observational data are good where you just see what happened, but we really had to then dig deeper and do what we call, randomized controlled trials to make sure that what we're giving is really having the effect that we think it's having.
That's great. Thank you. Let's turn to vaccines. I think a lot of us feel we're really beginning to see the light at the end of the tunnel with the emergence of these effective vaccines. Before I do, just a reminder to the audience, we're going to have quite a bit of time for Q&A. So please use the Q&A function. And you really do have access to some of the leading scientists in the world before you today. So here's your chance to ask your burning question. So Joel, let's talk about vaccines. People often comment on how quickly the COVID vaccines have been developed. Can you tell us a little bit about the science behind this? We've already established that everything that we've been learning about the virus and our reaction to it is built on decades of science. But let's focus in on the amazing accomplishment of coming up with vaccines within a year. How far back do we go for that?
How far back do we go? We go way back, but effectively, I think you can think about a maybe 20-year time span of basic research that led us to have the knowledge that we really needed when we needed it emergently. And so I think about it as really advances in understanding proteins and protein structure. Jennifer mentioned the spike protein. The spike protein is something that we knew almost immediately what it looked like, how it worked, and what to do about it to make it into a vaccine. And the reason for that is because of an advance called cryo-electron microscopy, which allows visualization of protein molecules, seeing their shape, seeing how they fold, seeing even how they move. Together with a project at the National Institutes of Health, understanding the MERS spike protein. The investigators working on the MERS spike protein, MERS is a virus that's similar to SARS-CoV-2. But the workers that were studying that wanted to understand the structure of that spike protein. And they made several discoveries that allowed them to understand what stabilizes the shape of that protein. Those discoveries were absolutely key to making these vaccines that work so well. They made that discovery not knowing whether it would ever have any practical benefit. I think the other, going back about 20 years, is the adenovirus vector It's so-called adenovirus 26, was basically developed as a platform for HIV vaccination. And it was the work that led up to it produced understanding of which genes in an adenovirus can you take out and cripple it so that it doesn't replicate and make anybody sick. And what can you put into an adenovirus to use it as a vaccine to deliver the DNA for the spike protein into our cells, so that our cells make the spike protein. Our immune responses then recognize that as a foreign molecule, an invader, and develop antibodies and T cell responses to it. So really, a lot of things were pulled together that weren't necessarily designed to deal with a pandemic in late 2019, early 2020, but the wisdom of people working on basic discovery, pulling those basic discoveries and understanding together in a big hurry, really accelerated the vaccine development process.
So how about tell us a little bit more about the 3 vaccines and in terms of their basic properties and how they're different, and of course the topic that's on everybody's mind, which is what's going on with the variants that are emerging and how effective these vaccines are going to be.
So the first 2 vaccines that were authorized for use in the United States, from Moderna and from Pfizer-BioNTech, are really pretty similar. They're certainly similar in their composition and then the sort of biological basis. They both contain the RNA coding for the virus SARS-CoV-2 spike protein. But RNA is a very fragile molecule. Jennifer's laboratory knows this very well, knows how to handle it, and knows how to not have it fall apart. But RNA really falls apart easily. There are enzymes that degrade RNA all over the place. So the RNA needed to be protected. And so it turns out that in both vaccines, the RNA is protected by something called a lipid nanoparticle. And again, some basic chemistry was involved in understanding how to make a lipid nanoparticle that protected the RNA just right, but released the RNA at just the right time inside the cell so that it could be coded, used to code for the protein. So those 2 are really quite similar. There's also some chemistry, some RNA chemistry that went behind it to make the RNA molecule itself not too inflammatory, because RNA inside of our cells, the kind of the wrong kind of RNA can be problematic. And so those 2 are really quite similar, and they've got modified RNA, they've got lipid nanoparticles as their delivery. Then the adenovirus vaccine has really got a different delivery system, and it also has the DNA rather than the RNA. So the adenovirus is a particle. Adenoviruses get into our cells like the SARS coronavirus does by binding to a specific protein and getting kind of taken up by the cell. And so the adenovirus vaccine uses an adenovirus coat to get the DNA for the SARS-CoV-2 spike protein into our cells. It needs to get into the nucleus, where then it's transcribed into RNA, translated into protein, and the same thing happens as with the RNA vaccines. Now, an often question is, why are we giving 2 doses of RNA vaccines and only 1 dose of the adenovirus vaccine? And the full explanation is that a little bit complicated. One explanation is, well, it's just the way the clinical trials were done. Another is that there are some theoretical issues having to do with immune responses to those adenovirus proteins that might make a second dose less effective. So when it was tried, it was just said, let's try it as a single dose, and it works really well as a single dose. I think the strategy for the RNA vaccines was a bit different because there were no RNA vaccines that have been used in humans. So I think the trials were designed, let's give them the best shot and find out if this is a principle that we can use for making vaccines at all. And it worked extremely well.
Mm.
All of those platforms are pretty adaptable with respect to making a new version with some of the viral variant sequences that are being discovered, and that, there's a lot of attention being paid to it. And Joe mentioned the importance of that.
Great, thanks. Well, let's move on then to other options beyond vaccines. Annie's already commented a bit on the testing of available therapeutics. Ashish, let's touch on ways that we could help our cells just protect themselves without necessarily having to develop, the antibodies that are part of a vaccine response. What's the promise of identifying and targeting reactions of our own cells to the virus when it's attempting to attack it? And of course, you've been key in developing a very, very interesting approach using nanobodies.
Yeah, great question, Dan. So, I like to think of kind of two bins of what we'll have in the future, to tackle either this pandemic, if the vaccines, continue to be effective, but perhaps, for future variants, but perhaps also for future coronaviruses or other viruses. So one general kind of very exciting idea is that instead of perhaps targeting the virus, what if we could target the machines in our body that the virus hijacks in order to do its business? But really, in order to have this idea work, one would have to have a really deep idea of how SARS coronavirus 2 or other viruses hijack the human body or human cells to replicate and do whatever it needs to do. So there's been a large effort both here at UCSF and at many other institutions to really map out what these interactions are. But the dream is that perhaps if there are common nodes or common things that many viruses use to hijack again the host, that perhaps you could target those specific nodes, and those would be good solutions both for the current coronavirus, but also for perhaps other viruses. And as a new pandemic, for example, comes up, perhaps if that virus shares a common node, then we would have things that are stockpiled and ready to go and perhaps effective. So that's a lofty strategy. There aren't great examples yet of clear molecules that really work effectively for that. But there's a lot of effort to find molecules like that or repurpose them from things that have been developed in the past for something else, and a lot of effort to really push that forward. Now, the other big bin of strategy is, okay, what if we could develop molecules that directly are very specific for the SARS coronavirus? And if we think back to the AIDS situation, there's this huge push to develop molecules that were very specific for the machines that AIDS or HIV uses to infect cells. And that's kind of, that process takes some time. It takes a lot of effort to, one, know what the machineries are. So as Jennifer mentioned, with structural biology now, we have a remarkable perspective of knowing what these machines look like. But on top of that, we have amazing new technologies to be able to go discover molecules that target those machines. There's a lot of effort to use these technologies to pick off each one of these targets that we think could be targeted by a drug, and perhaps that would be effective again for this virus and perhaps other coronaviruses. Now, you also mentioned other ideas to inactivate the virus, and one other broad idea is to use monoclonal antibodies or antibodies that we don't induce people to make, but that we could give to people to target infections. And Annie, alluded to this a little bit earlier. And these approaches we generally think are going to be very effective, perhaps for the very early stages of infection, or perhaps even as prophylactics. And one of the kind of distinguishing things that my lab and a few others across the world have been trying to do is figure out ways to provide these antibodies not through the blood, but perhaps through other routes that might be far easier for people to take. And if that works, perhaps people could take that at home, or self-administer these kinds of molecules, and that might change the course of this pandemic or potentially future pandemics.
So can you give us an update on where AeroNAbs is?
Yeah, there's a company that's licensed the molecule, and they're now doing the kind of, again, very hard work, but important work to gear up towards trying it out in patients. And at the same time, we're looking very closely at viral variants and testing both that initial molecule that we made, and then also developing new molecules that are going to be effective at, for example, the South Africa variant or the UK variant.
So, and Ashish, what do you think this will look like in the future? Since reading about your discovery, I've been imagining that people, would go to the pharmacy and get an inhaler of some sort and just do a little puff if they've been exposed. Is that what it'll be like with the next pandemic? If we can stand up this technology quickly?
Yeah, I mean, that's, we'd love to have it be safe enough and deployable enough that one could do that. I think that's a little bit of a lofty goal. One can dream, though, of having a broadly neutralizing nanobody that perhaps inactivates many coronaviruses. And certainly there's some precedent for this for influenza viruses. So nanobodies that really, neutralize many influenza viruses. And we could imagine that having that, ready to go and stockpiled, at least certainly for the next influenza pandemic, could make a pretty important dent, in how that evolves in the future.
Mm-hmm, mm-hmm. Let's come back to genomic technologies again, and Joe and Jennifer. The revolution in sequencing, the discovery of CRISPR and genome targeting technologies, is so evident. It's changed everything in science. How do you see them being applied in future outbreaks? It's kind of a softball question, I guess, right? We're just going to be that much capable of identifying what the next culprit looks like. Joe?
Sure.
Are there other technologies to make it even faster?
Yeah. Let me just back up and just think about where I was a year ago, January. I was actually in Cambodia, in Phnom Penh of all places, trying to set up basically sequencing centers in low and middle income countries around the world to be like an early warning radar for new and emerging viruses. I was a little late in getting that set up, obviously, but the concept that we could have a monitoring network of real-time molecular stations that would report out on viruses throughout the world in real time to be analyzed by the scientific community as a whole is totally within our grasp. I think the philosophical question here, Dan, is whether the world or any country is willing to pay for something to protect against something that might happen. And that's not totally clear. I mean, we can look back to the 1918 great influenza all the way till now. These things have happened before. They will likely happen again. But generally, our economies and our leadership are unwilling to spend large amounts of money and prepare for something where they're not really sure it'll ever happen. So that's a blocker. But we have the technology today to do this.
And Jennifer, your thoughts on that?
Right, I agree. I think, if there's a, there are many lessons from the pandemic, but my hope is that one of them is that we must be prepared for future pandemics. It's much worse economically, if nothing else, to not prepare for the likelihood of a pandemic. The question is, how do we do it? And I think Joe's strategy is certainly one approach. Ashish, your approach also could be very, very valuable. And in general, I just do think we need more vigilance in terms of how we monitor and detect infections, and then importantly, communicate about it so that there can be global preparations. I think for me personally, I think one of the I'm sort of shocked now to think back to a year ago about how and I think many other Americans, at least at the time, sort of thought that, this was a virus that was affecting people elsewhere, but it wasn't going to affect us, or at least not in a big way. And, how could I have thought that? I don't know, but I do think that in the future, having ways to communicate effectively about emerging viruses and get the word out quickly globally so that people can prepare will be critical.
Well, great. I do have a couple of other questions, but we've got some really interesting questions from our audience. So Jason, let me turn it over to you, and you can call through, and we'll call on our panel as appropriate.
Sure. So I have a number of questions from our audience. I'll direct some of the questions to specific speakers, but I encourage everyone to chime in if they have any thoughts on these questions. So I have one for Jennifer. Jennifer, what CRISPR-based technologies are being developed for CRISPR testing or for identifying other viruses? I'm sorry, for COVID testing or for identifying other viruses that might pose a threat to us? And do we have any idea of when those technologies might be ready and how useful they might be sort of more widely?
one of the interesting things about CRISPR is that in nature, it works as a surveillance system. It's a way that bacteria are monitoring themselves and their environment for viruses in real time and acquiring new immunity as they need to. And so I think that, using CRISPR in a diagnostic capacity for human viruses is very exciting. There are many groups working on this. I've been proud that we have a wonderful academic consortium here in the Bay Area that includes folks both at UCSF and UC Berkeley and the Gladstone who have been participating in this effort. And as a result of that, we now have 3 different CRISPR-based chemistries that are working for viral detection. And the goal of those efforts certainly has been to develop point-of-care testing, where you could, in principle, do surveillance monitoring of saliva samples, a very simple kind of assay that would allow people to monitor their own health and health in the workplace or in dormitories, et cetera. I think that's likely to happen. I also think that in the future, we'll see both at-home testing with CRISPR as well as laboratory-based tests with CRISPR that will, again, enable rapid, inexpensive testing that can be done that will allow us to, do the kind of surveillance that I think we're all becoming, aware is probably going to be a part of our lives for a while going forward.
Thank you. I have a question now for Joe. Joe, should we have foreseen COVID-19? And where is the next big threat in terms of diseases like this?
Well, wouldn't we all like to have the crystal ball about where the next one is? And no one has that crystal ball. But, I think that, we need to look back at history. There have been several big pandemics. And the coronavirus family was on our radar. I worked on the first coronavirus in 2003. That told us about the potential of coronaviruses that probably originate in bats like these did. And then we had MERS, right? Another separate coronavirus, also highlighting pandemic potential of this family. And of course, we had seasonal coronaviruses that were probably pandemic way back in history and then became part of our ecosystem. So all the signs were on the wall that this family had potential to be a pandemic. Now, a lot of folks have been thinking more about influenza because of its history in the 1918 pandemic and subsequent other outbreaks. But certainly the writing was on the wall for coronaviruses.
Do we expect the next big threat to come from coronaviruses? Or are there other viruses out there that we should be worried about?
Oh, there's lots of other viruses. Now, where's the next big one going to come from? I mean, I wish I knew. I think everybody on this panel wished they knew. But there's really no great way to predict other than to actually be vigilant and create systems that are going to let us watch how the viruses around us evolve and change and move from population or animal into population. And if we don't do that, we ignore the signs at our peril, and we just are doomed to repeat the mistakes of the past again.
Thank you.
If I could
Go ahead.
If I could just add a comment. I mean, I think we don't know what the next virus pandemic is going to be, but I think we can predict how it's going to travel, and that's by the airborne route. In other words, the massive success of SARS-CoV-2 is because it is so readily transmitted. SARS-1 and MERS really aren't that efficiently transmitted human to human. And so an infection that's transmitted by the respiratory route is really the best way for a pathogen to get around the world. And we're seeing that happen right now. So if we can understand what the respiratory viruses that are on the way are and what the categories are that they can come from, we can probably make some pretty good bets about where things are going to come from.
I'd like to
follow.
Go ahead.
HIV did a pretty good job.
Yeah, HIV did a pretty good job.
Just saying.
Without a doubt. But I'll also add the one surprising thing that really caught me off guard in this pandemic, and I'm sure a lot of other people too. I originally thought, hey, this is probably going to be like SARS-1. It's the same kind of genome. Anybody who's symptomatic has it. If you don't have symptoms, you don't have it. Guess what? Totally wrong. In this virus, 30 to 50%, depending on the study, and including our own studies, asymptomatic, never had a symptom, yet fully infected and transmissible. That feature alone totally changed the game and drives this pandemic in a way that makes it incredibly hard to control.
And I think adding to that, it's humiliating to think about the fact that we don't know why it's so transmissible during the asymptomatic infection compared with SARS-1. That's something we don't understand, and we need to understand it.
And just related to that then, if this had happened 20 years ago, where would we be a year into this pandemic?
We wouldn't have the 3 vaccines we have right now. We might have made a vaccine, but it probably wouldn't have had the protein conformation that has been idealized, and it would have taken much longer to get it done. We'd be in really, really deep trouble.
That's almost
We have-
certainly true for all the monoclonal antibodies too. I mean, that's really resting on, original work on the original SARS virus.
And same thing goes with clinical trials. We're really piggybacking on existing clinical trial infrastructure so that people can get things up and going very quickly. Some of the medications are repurposed. Remdesivir was originally an Ebola drug, and a lot of the antivirals that have been developed and looked at were developed for HIV or viral hepatitis, and I think really are building on the back of work that we've done so far. And I think one lesson I think we can certainly take away from this is that a siloed approach to medicine where we say, well, this is just what I do and I don't do anything else. And, there were no COVID experts a year ago. And if we had waited for that field to mature, we wouldn't have gotten very far. So I think it's really forced us to all think about flexibility and leveraging the existing infrastructure, which is why we're hopefully better off now than we would have been 20 years ago had this come to pass, because people were able to shift their expertise.
Yeah, and if I can just comment on that too, because I think the other thing that's been part of that whole, building that whole ecosystem of people jumping in who didn't necessarily have expertise in virology or anything seemingly relevant to COVID, who have been able to contribute, is the fact that that information is getting posted very quickly to preprint servers. We've seen medRxiv making articles available, and they're cited on PubMed, so you can find them quickly. And even though they're not peer reviewed, and so you have to take that into account when you read those papers, it's been a great way, I think, to disseminate information very quickly, publicly. It's available to anybody that wants to read it. And it really, I think, has driven the field quickly in a way that we haven't seen previously.
Great, thank you all. Another question that we got is, would it be possible to create a platform to build vaccines for coronaviruses or perhaps other viruses so that we can actually inoculate people ahead of time? Would we need, what kind of monitoring and what kind of technology would we need in place to achieve this?
Well, actually, that's an interesting point. Maybe I'll make a comment, and then I think Joel probably has something to say on this too. That one of the silver linings of this pandemic may be the fact that we've accelerated our vaccine design technology so fast, mRNA vaccines being a prime example. And so is it now the time to think about those technologies for things that have suffered for lack of a vaccine for so long? I'm thinking diseases like malaria, tuberculosis, Chagas disease, syphilis, many, many others that could easily be a target of mRNA vaccines that really just have not moved the needle in so many years. Maybe that's going to be a consequence of all these new advancements.
Joel?
Could very well be. I think I agree with Joe. I think another thought is just it may be difficult to know exactly what to predict. Vaccines and the immune response are pretty specific. And so you really want to develop a vaccine strategy that can be implemented quickly, but probably not too far in advance. If it could be very tightly coupled with the kind of surveillance system that Joe described, so that if we see a signal, let's just say it's coming from Joe's station in Cambodia, and we see that there's local spread, there's community spread, it seems to be respiratory transmission, that sort of thing should trigger a fairly well-designed series of events that have already been discussed in advance, planned in advance, so that a vaccine can be designed and implemented even more quickly than it was for SARS-CoV-2.
Wonderful. I'll direct this question first to Ashish, because I know you are working on, or at least considering how to deal with the rise of variants in therapeutics, but I invite everyone else to contribute as well. So we had a question where someone asked if they're curious to know if there's a limitation on the number of mutations that the spike protein or the virus can ultimately have, and if some variants are already approaching the endpoint.
I could certainly tackle that from the context of, therapeutics and, how we would think about developing, for example, alternative antibody-based therapeutics for the variants. I mean, that's basically a cat and mouse game. So, a variant arises and we can try to find an antibody that basically catches up. We're thinking maybe more broadly about antibodies that that could be broadly neutralizing. And that's, again, only for antibodies that target the spike protein. There's, therapeutics that could target a whole host of other viral machineries, and typically those don't drift to the same extent or change to the same extent that the spike protein does. But at the risk of cold calling Joe, I'd say that Joe is probably far more knowledgeable about viral evolution and thinking about whether it's a true endpoint. I would venture maybe not, but maybe Joe could opine on it.
I don't think we can really calculate what the limit of diversity is. And my guess is it's very, very large. Protein space, and Jennifer can comment on this too, is huge. It's astronomically large. And therefore the opportunity to the virus to evolve and change is really there. But it only has as many chances to evolve as we let it. So the faster we get immunizations and vaccines out, the less chance that occurs.
I will comment, there is a system that's been developed for expressing the spike protein receptor binding domain on the surface of yeast, and then screening and then making a mutant library, and screening all of those for the ability to bind the receptor ACE2 molecule and the ability to be recognized by antibodies. And that's given at least some information on the limits of mutational tolerance. And so that's at least helped. You gotta know what to watch for, theoretically. And so there's a bit of information, but like Joe said, I mean, it's an immense world. And I think I think of it as, if we can combine our efforts to drop the global burden of viral particles, then the likelihood of new variants decreases as we decrease the number of viral particles that are out there mutating.
Great. We have a number of clinical questions that I'd like to address one to Annie. This one is about asymptomatic infections. If asymptomatic infections in fully vaccinated individuals turn out to be a reasonable possibility, how likely is it that a vaccinated person with asymptomatic infections could later turn out to have long hauler symptoms?
Yeah, so this is a great question that, as with most of these questions, we don't have the answer yet. And I want to answer a couple pieces that we do have the answer that addresses this. So one thing is that we know that people can develop long hauler syndrome without having symptoms initially. So that's a little bit distressing, but we have seen, I think, reasonable data coming out recently to support that. So certainly we can see it in people who've been seriously ill, but also in those who have been asymptomatic. My guess is, and this is just a guess, but that what we're going to find out about asymptomatic infection, if and when it occurs after vaccination, in people who really have just a little bit of virus around, is that it's probably not going to be sufficient to really set up a robust infection or inflammatory response that's probably going to be responsible for leading to long haul syndrome. Whether or not that's infectious to others remains to be seen. And again, we're learning a lot about this, but I suspect we are not going to see long haul complications. And indeed, we've had some intriguing data that even vaccination may be associated with the reduction in long haul symptoms in some, although we need to learn more about that. But I want to distinguish that from vaccine failures. I think that we know, as we're learning, most of these vaccines look like they work very well. to prevent serious disease and hospitalization. But some people in clinical trials have developed symptomatic disease. And that is a sign that the vaccine isn't working quite as well as we want it. We'd love for it to prevent all disease. So developing symptomatic disease, you know, what the risks of having long haul syndrome after that, I think we're in the dark as to what that will look like. But I think that's a very different syndrome from what I expect will be very limited viral shedding from people post an effective vaccine. But a lot of that is speculation. And any of the other panelists, feel free to take me on that one if you disagree.
I agree with you.
Thank you.
We seem to have no takers on the challenge. I do have another question related to vaccine therapies. And do we know how long the current vaccines provide protection for? Is it likely we'll need ongoing booster shots? And what kind of trials need to be taken in order to determine how long these vaccines will last?
So I think that's a really important question. And I think that the data obviously are incomplete because we haven't had these vaccines for very long. But the good news is, in the time that we have had them and been able to study them, we haven't really seen a significant waning of the levels of immunity. There was a little bit of concern early on in that there were some observations that said that the viral anti-antibodies to the virus peaked early and then they decreased. And that was a concern, but that's what the immune response does. In other words, it generates a flurry of responses and then it kind of tapers down to a steady level. Right now, it looks like the half-life of the antibodies after vaccination are at least a year, probably longer than that. What we don't know yet is exactly what level of antibodies is sufficient to protect. And that's where the variants might come in. So there's a lot of monitoring right now, an increasing amount of viral sequencing to know what variants are arising, and a lot of attention given to, you know, are the antibodies and T-cell responses induced by vaccination beginning to wane, or are there breakthrough infections and is it time to modify the spike protein version in the vaccines? So there's very close surveillance of that very question.
Thanks, Joel. And Jason, since we have 5 minutes left, I'll have a wrap-up conversation with our panelists. So everyone is wondering what the future holds. And our discussion has been focusing a lot on that. And we'll also agree that it's a fool's errand to try to predict the future. But I did want to focus in on two things. The first one is, do you think that the way of doing science has been inextricably changed going forward? In other words, will we all look back and say, you know, back in 2020, you know, there was a shift in the paradigm of doing research? Yeah, I'm seeing nods, yes.
Yeah, I mean, without a doubt, like Jennifer mentioned, for example, the preprint servers. It was just a couple of years ago, I would mention preprint servers and people would be like, what's that? Why should I care about that? Now everybody knows because it is the way we can transmit information so quickly when we need it the most, with the caveats that Jennifer mentions. That's one obvious way.
Yeah, and I think for clinical trials, in a good way, things have been really accelerated. We've seen barriers that it has taken us years to try to get broken down in terms of consenting people remotely, not having monitors come to campus. I got a clinical trial up and going in 11 days, which has never happened. And I really hope that will continue. And I think that we all have been able to really say, look, this is an emergency. Let's cut out some of the stuff that's getting in the way, and then we can re-envision what do we really need. But I think the level of collaboration that's out there has been remarkable, and the ability to, and I want to be clear, I don't think any of it's been shortcutting anything around safety or good science. It's more been wow, do we really need to wait for a conference to present the data if it's life-saving data? I don't think we're going to do that anymore. I mean, conferences are important, but we used to say, well, we're going to wait for that conference. And so all of those things, I think, are a good way to kind of rebuild and say, well, what are what are the things that we're happy to let go of? There are a lot of things from the pre-COVID world I'm happy to let go of.
So again, just because we have a few minutes, the harder question about the future, and that is for the billions of people on the planet who don't do science for a living. What will life be like for us in 3 or 4 years? Will we be back to going to concerts, packing airplanes, not having to worry about masks? Will life return pretty much to the way it was before COVID came along?
That's pretty philosophical, but I think it won't ever be the very same. I think, people talk about the new normal. I don't know what that means, but I think life's going to be different, but life's going to certainly return to the point where we can see other people, see our friends, see live music, and not fear for our lives. But I think people will take precautions. People will be aware. Maybe restaurants won't be so crowded and noisy.
I think we're all going to view masks differently too. I was struck right before COVID hit. I had a bad cold and of course still came to work, but I wore a mask and I felt like it was really weird to do that, but I did it. I wasn't seeing patients, but I think we're going to learn that masks are a really great tool to have. And I think there's parts of the world where they use masks very comfortably before this all happened. I think we're going to think about our travel a little bit more thoughtfully. I think we're going to think about our relationship with the animal world and massive animal husbandry and that as a resource, as a reservoir of potential infections. I think those are all good things to be thoughtful about. I think we'll also really look at all the wonderful vaccines we do have and not take those for granted, because we have life-saving vaccines that are available right now that we aren't reaching people for a whole variety of reasons. And I really hope that we can re-envision a lot of that as a good part of our new normal, because I think there are things that should change and can change as a result of this that will be tremendously positive.
Okay, well, we've reached the top of the hour. Thank you for just a delightful conversation. I want to remind everyone that this is the first of a series of 3 presentations on COVID-19, the path forward. And the next session will be the mandate for equity, which is going to be on Tuesday, March 16th, same time. And then the last in the series is mapping a post-pandemic world, which will be on Tuesday, March 23rd. And again, thank you for attending and join me in the appreciation we have for our esteemed panel. Thank you for a great conversation.